here at the University of Guelph for the Biodiversity Institute of Ontario. The goal is to catalog all the biodiversity on the planet. It's exciting because of what it means to the world.
I think that DNA barcoding is such an amazing new technology. I've been working at Bio since 2006, and just to see how everything, where it started and where it is now, is just amazing.
DNA barcoding is a method to identify species.
In order to accomplish that, we help people collect data and we do development in order to gain more insights into these big data that people have gathered all around the world.
What I do is I array all the specimens into 96 well microplates, and there's a ton of insects in each of our samples. And DNA barcoding is extremely awesome because I cannot tell myself what everything is in that sample. DNA barcoding, we just all put it in the plates and it does the trick for us. It'll tell us what that species is exactly. What I really love about DNA barcoding is such a cool tool for helping to identify species way faster than we have in the past.
The downstream goal is to actually have a handheld device so that scientists, researchers, field workers go into the field, out in the field, and basically analyze a sample on the spot to know more information about the sample that they've recovered.
The most amazing things were always when you discover something new, a species you would not have thought of has been not encountered before. And a lot of times it's actually, for example, fishes that people have been seeing for years and years. They all look very much alike, and then it turns out with the help of DNA that a fish that looks always the same is actually 2 or 3 more species.
I think what we differ from other people is that we not only provide the lab, the collection team, and also the informatics support, we also provide them insights about the data because we care about the data, so we actually have teams that are focusing on helping the user going from the beginning of their research all the way to the end of publication. So I think that's really the main big difference between us and other people.
It's building that reference library. So having a lot of DNA sequences for everybody available on the planet through an online database.
The Biodiversity Institute stands out among other facilities around the world because we're a high throughput facility. We deal with tons of specimens.
We have gotten through a lot of samples that are in upwards of 100 years old and have been able to process and get great results. That's the most exciting thing to me is, coming from a background of high throughput genomics, is that we're constantly pushing the envelope for new technologies.
I don't think there's any other facilities around the world that are as fast and high throughput as ourselves.
What we're doing has some relevance to society. It's more than just my own curiosity. It is something that will help society control things better, avoid certain things, so it really makes a difference. Hello, and welcome to the Biodiversity Institute of Ontario. My name is Dirk Steinke. I guess you've seen me once or twice in that video before. I hope you had a chance to actually watch it, which would mean that technically everything is fine. So, yeah, what I'm trying to do in the next hour is present you a couple of things. I know you all have your Malaise traps already up and running. You changed the bottle already, so you're well into week 2. I really hope the first bottle had enough insects to actually give you as many barcodes as we can probably run for you. So what I'd like to do is present a little bit more of what we're doing, put that Malaise trap some sort of a context. So what are we doing with it? Aside from giving it to 60 different schools in Canada and have them collect insects for just 2 weeks. But I also like to show you all the other trap methods we're using for that, because the Malaise trap is just one in a collection of traps we're using for the entire summer season. So the first thing I'm going to do is actually present you a couple of those traps and ending up with a Malaise trap just to show you that we have set up one here, explain a few things, although I'm pretty sure you're all professionals by now because you have it already for a while, you help pitching it and all sorts of things. So I will not spend too much time on the trap itself, but on the other ones we're looking at. One Once that is done, you'll see another video that introduced you a bit to DNA barcoding. I know there's also classroom materials where you had a video you can go through a PowerPoint presentation with somebody. Barcoding is all about, what's happening in our lab. After that, I'll take some time to introduce you to a couple of groups of insects you're likely going to find in your traps. So just to introduce a couple of those species that are part of different groups of insects, they probably have a few interesting stories to tell. And when I'm done with that, I'm going to take the camera and show you a little bit around in the BioBus, so the field vehicle we have for that kind of work we do in the summer. So that will fill pretty much at the end, which we like to use for your questions. So you can ask me questions, somebody types them in, and I can read them out loud and answer them for everybody to hear. Again, that's taken in total about an hour. So I better start going now. So as I said, I try to start with the traps we're using in the field. So the first one is right behind me. So you shouldn't focus too much on the institute behind, but on this trap here. And it's very similar to your Malaise trap. All this tent stuff around it is not there. The principle is the same. This is an intercept trap, and actually the Malaise trap is an intercept trap as well, which means you intercept the flight of an insect. So it flies, it buzzes around, bumps into this mesh here, and in this case, most of them, the bigger ones we're after, will fall down. In the Malaise trap, it's a little different. There we are after these insects that by insect trying to fly around that thing and go upwards. So when they fall down here, they fall in one of those trays. We have two of them out here. One is just to show how we buy them, but usually we don't leave them as black as they are. We paint them yellow. There's a reason for that. I'll talk about that in a minute. But what we will do is, underneath that trap is fill it with water, put a few drops of soap in them, because if we wouldn't do that, and we just have this thing filled with water, a lot of the insects that are caught through that trap and bounce here down in the water are light enough that they can actually walk on the surface of the water and walk away. And that has a lot to do with the physical property of surface tension. So water has a surface tension that allows these animals to crawl in like a water strider. If you put a little bit of soap on there, and that's the idea of dishwashing as well, they drown. So they cannot stand on top of the water, so they will drown, sink in. And that's how we can collect them. Unfortunately, we have to use water for these kinds of traps. They're huge. You couldn't just use another liquid like the ethanol we're using in the Malaise trap, because if you have a trap like that big, you would have to use a lot of ethanol. Then if you have a hot summer day, ethanol has a problem. It evaporates much quicker than water does. So the trap would be empty within an hour. So then you can't use it at all. Plus, ethanol that wide open in a thing that looks like a food tray is probably not such a good idea when big wildlife is around, probably drinks it. After all, ethanol is a poison. So we use water for that. Comes with the problem, we'll have to collect or go back to these traps pretty much every day. So they need constant service. And that's a bit of a drawback, and that puts the Malaise trap certainly an advantage to that one, because there you don't have to come back all the time. So far so good, flying insects, but we probably want to collect something else as well. So if we are out in the field, we want to not only get these flying insects, big ones for this kind of trap, smaller and tiny ones that fly for the Malaise trap, get to that, but what about the ones that crawl on the surface? Everything that's crawling around here usually doesn't fly a lot. So what you can do is, relatively simply, you build a pitfall trap, and you see one here. And I grabbed a little container from it. So this is nothing else but a small yogurt, sour cream container cleaned up. So everybody could build one of those pretty quickly. We fill it with one of our preservation liquids, usually it's ethanol as well, and then put it in the ground. So what you do is you dig a hole and that it can go in and is level with the surface. So any insect that crawls will crawl here and fall into that. So a lot of spiders, not only insects, are trapped in that kind of trap. The problem is if you have ethanol like that, and you would leave it without any cover, any rainy day would probably fill up that thing pretty quickly, and nobody would be trapped in there. and the ethanol would dilute more and more, which makes it less efficient as a preservative. So we built little roofs out of a styrofoam plate and 2 skewers, and that's pretty much it. And then we can leave those traps alone for a day or 2. Same problem, because they're relatively exposed, you have to come back to them. So again, a type of trap where you have to come back over and over. So that's why we don't use them for an entire season. We go to a place, use these traps for a couple of days, a week or so, then we pack them together and go to the next site, while we leave the Malaise trap behind for the entire season. So far I've been looking at animals that are active a day. Even your Malaise trap only rarely collects night active insects. A lot of the moths that are out there at night are usually not caught in there because their orientation and navigation system is so sophisticated that they will track down a Malaise trap before they even approach it and then just simply fly around it. They have to do that because they are flying out at night. There's no light source for them, so they have to have a sophisticated system to find any kind of obstacle. So we're not in a position to fool them a lot. So if you want to catch those, we know they are attracted by light. We know that. You just simply have to look at a street light at night and you see how many bugs circle around it. So what we do, for example, I haven't shown that here, but it's very simple. You take a white sheet, something like a bed linen, and span it out like between 2 poles like this intercept trap. But instead of just leaving it there, we take a light source and shine it against the white surface. What happens is that after a few minutes, tons of bugs will land there because they're attracted by the light, and you can simply go there and handpick every single one of them. That's, as opposed to most of the other traps I show you here, it's a very selective trap. So you can go in and say, okay, I'm interested only in this particular moth, and I pick that, or that species, and I pick only representatives of that, and leave the others alone. While all the other traps, they just collect whatever is trapped in there. And we do the sorting at the end, where they're usually killed in the jars. So another light trap, which is also not so selective, but pretty good, is this bucket trap. So it's essentially a plastic bucket, then it has a little lid, and a plexiglass structure that holds a circular light bulb. See that here is a relatively common cold light thing. This one is connected to a car battery, and the car battery has enough power to run that thing for 1 night. And then the next day we just have to recharge the battery and run it again. What happens is the bugs are attracted to this light source, and then let me remove that. They fall down in this funnel here, or let's say here on the edge of it, but because it's smooth, they just slide in and go through this opening in the bucket. And most of them do not manage to come back up again. It's a bit too much for them to find the actual exit. Plus there's a little bowl in there with some sort of a preservation and killing agent like ethanol or something like cyanide. So they're trapped in here, and for us it's relatively convenient. The only thing we have to do is at the end of the day set that trap up, connect it to the battery, come back the next morning and collect everything that got trapped in the bucket. So another light trap version, a bit more advanced than, let's say, the sheet. Believe it or not, also a very common trapping method is the sweep net. Not like in the cartoons where somebody is chasing one single butterfly for hours and hours and trying to catch it. That's not the idea of these traps. You can try that, be my guest, but this is not usually how we do that. It's very often used if you go through areas where there's high grass or anything, like knee-high or even higher, and then you sweep back and forth. And while doing that, you collect everything that is actually usually holding on to these grasses and everything else, and then later on you can start moving them all out. So with that, you collect a lot of insects, a lot of different spiders also. Still very effective. A lot of scientists use that when they go out in the field. Usually as a first one, so they're trying to sweep and check out what is around there before they start with more sophisticated methods. Together with the last thing, the Malaise trap, we actually use all these methods. We call that a standardized protocol, which means we use all of them for a certain defined period of time, a number of times throughout several days. at certain defined positions. The idea of this standardization is pretty simple, and you do that a lot of in science, is you can redo the same experiment at different places, but you can compare the results because you've always done sort of the same thing. If you would change your methods or to use 2 traps here and the other 3 there, you would not be able to connect and also compare whatever you collected in the 2 of them. So you always have to do sort of the same thing, and that's what we call standardized sampling. And all these traps are part of it when we head out in the summer and collect. And the Malaise trap is part of that as well, but this one, as such, is the only trap we use that stays at a particular space. Summer means starting in May, ending up around this time in September, end of September. That's our field season. The rest is usually Canadian winter, which means there's not a lot of activities of arthropods, so insects, spiders, and so on. They're not usually around out there. So we use these traps for that kind of season. We have colleagues in the world that don't have these kind of seasonal changes. So we have Malaise traps that are actually active, 365 days of the year, because they're somewhere in the tropics or in warm California or whatever. And you can imagine there's much more we catch through that. But here in Canada, on average, it's really just the May to September season. That's where we collect most of it. And while the Malaise trap sits in a particular park or several parks, all the other methods travel with our field vehicle from spot to spot, and the people just operate them for a few days, start packing up, going to the next site, and so on. And that's the summer. I'll tell you a little bit about the BioBus and how that works in a minute. So those were the trapping methods. There's certainly more, but they're either not part of our standardized sampling, or they catch something completely different, which is not part of what we doing today in this kind of virtual classroom. We should talk about other things probably in a different environment. So next thing is going to be a short video about DNA barcoding and such, how it works, what the principle is. And when we come back, I'll have the insect boxes for you, where we have grouped a couple of insect groups together to show you what you would can expect in such a catch. And the good thing is probably a lot of you have already had a peek in the bottle of the last week, so you can see and get my explanation of what you likely saw there for most of the part. All right, see you in 3-4 minutes. All right, welcome back. Time for a little show and tell. Don't worry if you can't see every single bug from what you're looking at right now. We have a couple of slides that will come in and show you the animals a bit closer, and I think you can actually enlarge them in the system as well. So what I'm trying to do is with this box, and there's another one that I will put on top of it as soon as we're at that point, is introduce you to some of the major, what we call orders in the insect group. And especially the orders where we think you're likely going to have at least one representative on you or in your trap, but also some others where I would tell you, okay, it's not likely that you find them because of several reasons. What you're looking at is actually representatives of entire North America. So not everything I'm going to show you is actually originally Canadian. In some cases, you might be even sort of relieved to hear that. Because I'm trying to pick on some stories that are maybe a little more cool or whatever you want to call that. So the first group I'd like to start with, and these compartments in the box are sort of arranged in the same way, is the Hymenoptera. So the order Hymenoptera, or skin wings, comprises of ants, bees, wasps. So very well-known animals, and we picked a few representatives that have interesting stories to tell. One of them is certainly the large group of what we call parasitic or parasitoid wasps. They're interesting because of their lifestyle. Some of them are extremely tiny. So in this box here, and I don't even if I point to it, you won't be able to see it because it's probably as big as a grain of dust. And it's a fully grown animal. It's a small parasitic wasp. And then there's a big one here. There's much more to tell about this guy here. It's called a tarantula hawk. So the tarantula hawk has an interesting reproduction strategy. What this animal does, and the name gives it away, is hunting tarantulas. So we're talking about animals that are probably as big as my hand. It will sting it. With stinging it, it releases a paralyzing toxin. So the prey is not dead, but it's paralyzed for the rest of whatever life it has. And the next thing this animal will do, the tarantula hawk, is carrying that paralyzed tarantula in a safe spot, burrow a hole somewhere or on the leaves, and lays an egg inside the spider and then goes. What happens is that the egg will hatch, the larva comes out, and then that one will start eating up the tarantula from the inside out. And once it's done, it's fully grown, it can go for metamorphosis and emerge as a fully grown tarantula hawk out of the spider. So that's what most parasitic wasps do, but depending on their size and their lifestyle and where they occur, they have different victims for that. So the tiny speckle of dust I talked initially about will go after a very tiny fly, and so on. There are 10,000s of species of these animals, and they're very specialized. They usually have just one type of victim, which is some sort of another arthropod, and usually not a vertebrate like us. So they have not developed a strategy to put their eggs in us. That's different animals, actually. A few other of the same group are, for example, the little bumblebees. They're usually very good indicators that there's really spring coming, simply because they are among the first ones active. Their fur, or whatever you want to call that, furry coat, enables them to come out when the temperatures are still very low, but they sort of have a good sense of when spring is around. What you see is queens looking for a new place to found their new colony. And the other ones are the wasps. I'm pretty sure you will have quite a few so-called paper wasps in your traps, because that time of the year they get increasingly active, sort of preparing the hive. Some of them actually are sort of desperate. to get a last bit of food. And those are the paper wasps you know. They cruise around you when you have something sweet with you, or they are very common around garbage bins. And the name paper wasp comes from the fact that they build a nest out of something which is pretty much like paper, and they produce that themselves. Another animal in here is the hornet, which is essentially a large wasp. I know there's tons of legends out there that so many stings of a hornet can kill you, which is sort of not true. It depends on how sensitive you actually are to all these toxins they release in self-defense. Still, the most deadliest among them is the one from the honeybee, and that only if somebody is actually allergic to this toxin from the bee, and that can actually really be life-threatening. But other than that, it really depends on your constitution and any existing allergies or not. Normally, hornets are pretty harmless animals. They just want to be left alone. Hymenoptera, you'll find quite a few in your Malaise trap. It's not the most abundant, so not the most specimens in the trap, but it's certainly among the larger groups in your catch. Let's move on to the other box underneath, which contains what we call true bugs. The problem with the name, nobody, not even the experts, can really explain why true bugs, not just bugs. I tried to check the literature. Nobody came up with a reasonable explanation for that. True bugs contain animals you know, for example, as stink bugs, bed bugs. I don't know if you heard of them. It's also, some people know them, assassin bugs. So assassin gives it away, so they hunt other animals in a very particular fashion. The stink bugs releasing a stinky substance when they are attacked in order to get rid of you. Most of those, exception assassin bugs so far, are plant eaters, so meaning they have developed a mouth that is shaped like a syringe, with which they can poke through a plant and then suck out the liquids. However, we have a question already. Let me stop at this part of the bugs. What types of bugs can be trapped during the winter here in Canada? Really, in winter? With the methods we're using, I think none, single one. Not a lot of them are active. What they need is higher temperatures somehow, so they have to, the only thing you could probably do is, if it's really a nice snow cover, you can dig underneath, and you probably find a couple of ants that are, there's one particular group of ants that are very well adapted to our winter temperatures, so they will hide there. And then there's also a famous animal called the thermometer cricket that is around for a longer period of time because they like the cold. Let me continue with the more aggressive part of the true bugs, which are the ones that actually have developed the same mouth parts, but they're not using it to sting into a plant. They sting into something else, another insect or other animals. And then they release an enzyme, so a substance that can actually break down proteins and other things. So what they do is they release this enzyme into their victim, which then sort of liquefied from the inside so they can suck it empty. A very prominent example, and very frequently here in our region, and a lot across Canada actually, is the giant water bug. Not only is he a predator, very aggressive, it hunts not only insect larvae, no, it goes after little tadpoles, little fish. It has been seen, and I think there's a video out on YouTube where this thing actually caught a snake. So a small, whatever, that long snake. Yeah, their bite is pretty painful. I got bitten by it once. It's pretty excruciating, so you don't want to mess with them. It's not that they attempt to suck you and get your stuff, your whatever liquids, body liquids to eat on them. It's just their defense mechanism. The first thing they do is going to sting you, and this is pretty painful. And that also has to do with the substances they automatically release at that moment. Another interesting group, and you can hear it actually these days, especially in Ontario right now, are the cicadas. So today, not now because there's a bit of wind picking up, the cicadas sang today. So it's a high-pitched sound that's in the air late summer, early spring, where they're trying to attract their mate. What they do is rub their hind wings against each other and do that in an incredible speed so that you can actually hear a high-pitched sound coming from that. So cicadas, very interesting animals. Shoebox, it's similar to the Hymenoptera. Very frequent in your Malaise trap. I'm pretty sure they're already in there, but not the most abundant one. That'll come a little bit later. Beetles are not that often, only if they're relatively small. The bigger guys are just too big. They're more like victims of the first trap I showed you today, the interceptor. So a big beetle would fly into it and drop down into the tray. And the other ones, in a Malaise trap, it's the same thing. They drop down, they just walk away. That's one reason why some people actually take a Malaise trap and take these pans, we have there, the yellow one, and put it underneath, on both sides of the mesh, so that you sort of combine the two trap technologies into one new trap that is even more sophisticated. So one beetle I wanted to point out is the diving beetle. I know there's a question, but I get through the beetles, then I answer the question. This dive beetle is an interesting guy. It's again one insect that hunts underwater and is not shy to hunt even vertebrates like fish or tadpoles. The dive beetle has an interesting way of doing his business and going down there, because his problem is, as ours is as well, when we dive, we need to carry air around. We take scuba tanks if we want to stay there longer. He doesn't have a scuba tank, but he has the ability to take water from the atmosphere, so he swims up to the surface, collects an air bubble and stores it under his protective wings or under his belly, and then he dives down and breathes through that. So it's sort of a little scuba diver. That also, again, it's a predator, and some of those guys can get pretty large, probably as big as the giant water bug, so you can imagine that they can go after pretty big prey as well. All of the other beetles are no-shows for our Malaise trap, but after all, it's the largest group of insects, about 200,000 species on the planet. Let me check the question. How long does it take to barcode a single bug? Oh, I had that question today in another virtual classroom as well. Depends on how quick you want to have it done. So regularly when we do our business here, about a week. And that goes from the bug arriving here in the house, somebody taking a photo of it, storing it like here on pins, getting a piece of it that goes for DNA extraction, and then all the steps in the laboratory to actually read the sequence, all the way to the point where we upload it to our database. So about a week. If there's any kind of emergency situations where we need quicker, we can do it within a day. And because the Malaise trap program, just to make sure you don't expect sequences within a week, there's so many schools participating that this takes a longer than just 1 week to go through all the samples you guys will send us. But on average, it's about a week. Another group, not very often in our traps, are the grasshoppers and crickets. I've seen in the blog we have up on our Malaise program website that there was one school who actually called it a pretty big grasshopper. I have no idea how that happened, because they're usually not coming up there. They can fly, they can jump, but they are very good in avoiding these kinds of obstacles pretty well. So I don't know how it happened, but it's very rare that you find some. Some of the smaller ones maybe, but not the big ones that are as big as your finger or so. So the grasshoppers. And now box number 2. What you can see here is the second largest insect order, the Lepidoptera, or moths and butterflies. Again, not so common in Malaise traps. You might find an occasional moth in there, smaller ones for sure. But I told you before, they are pretty good by flying at night. So they are far too smart and have too good of orientation systems that they actually are not collected very often. And the butterflies that are out, that they are usually either too large, or, especially this time of the year, if you checked around, I'm pretty sure you haven't seen a lot of them. So many of them are either dead or are already starting some sort of a migration. Some of them are very migratory. So chances are that you won't find any of those in the Malaise trap bottles. But this one, that's the big group, the Diptera or flies. The flies are the most abundant. Every trap we have somewhere on this planet will have mostly flies. And in most cases, more than 50% of what we have in the bottles there are flies, some sort or the other. It's not the largest group of insects, but still they're probably the most abundant, meaning there's the most number of individuals per species. So all our catches are dominated by the Diptera, and they have also the size that is perfect for Malaise trapping. Flies have lots of different lifestyles adapted to. Some of them are pretty good in mimicking other predators to have a safe life, like the hoverflies often look like a wasp or anything else. So extremely dangerous, but they aren't. But that allows them to be very, safe. And there is another question. Is that true? Okay, let me see what it is. Scientists have noticed a decline in the honeybee population recently. Has the Biodiversity Institute done any research on this issue? Not we, but colleagues here at the University of Guelph. We have actually a bee institute here, so we have a couple of people that are far more smarter in terms of what bee science and work with. And I believe not long ago they were part of a larger study that tried to figure out what actually kills all these bees, because it's a pretty dramatic decline actually. And it seems to be a mixture of several factors: changing environment, pesticides that are put out to protect our crops from other insects, in addition to certain fungi that are out in the air. So it's a complete colony collapse, they call that. So that means they're not just in a decline. When this kind of syndrome affects a bee colony, it's completely wiped out. Because what happens is that some bees don't come back and show where the honey is and all that. Pretty sad story. We have a couple of researchers here at the university that are part of that research. So back to my little flies. I told you about some that can camouflage, and then let's talk about the ones that have developed different ways to get after what is very dear to us, our blood. Two different strategies. So let's look at number one, most common, most prominent, mosquitoes. Mosquitoes have a changed mouth. Their mouth turned into a long syringe with which they can go through your skin and start sucking out your blood. While they're doing that, they're releasing a mixture of chemicals. One numbs your skin so that you don't feel the sting. The second one, because the sting is so thin, and you probably have noticed that if you take a thinner straw to suck out your pop, it's much more difficult than taking one that's a bit wider. So for them, there's a huge problem with really, really thin. What they do is they release another chemical that actually liquefies our blood so that they can suck it up easier. That's the mosquito strategy. Other insects are not that, further developed. So what they do, they use their mandibles, which is their jaws, to bite a chunk of flesh out of you. So let's take a very common one here in Canada. That's the black fly. Tiny thing, but still that's the same business. It has also these chemicals because it tries to avoid that you notice you've been bitten. So that's one liquid that is released so that you don't feel that you've been bitten. And once they bit the chunk of flesh out of you, they start licking the blood that comes out. And again, they make it more liquid so that everything goes quicker. Normally, you don't notice that you've been bitten by a black fly. Long after it has left, you start scratching and wondering why the blood keeps flowing and flowing. This is because of chemical number 2 that actually makes your blood thinner. Larger cousins do the same thing, and larger cousins I mean with the not the hoverflies, the horseflies and deer flies. Their bite, despite any chemical thing, hurts, and but they're usually slow enough that you can notice them before they actually start their business. So flies, extremely abundant in your Malaise trap. Rarer ones are in the other 2 boxes, and I'll go quickly through them. You won't find any praying mantis. I guess 2 weeks are not enough for that slow animal to crawl up there, and they have no interest in actually going somewhere like that. Same as for cockroaches who try to hide somewhere where it's dark. You'd rather find them somewhere in a house, in a basement, or wherever there's food close by. That's where they like to go. One group I'd like to talk about just briefly, just to do them some justice, are the earwigs. their name earwig is really bad because it has nothing to do with what they do. Earwigs don't crawl in other people's ears. Their pinchers in the back don't pinch you. All that kind of stuff is nonsense. So they got a very bad reputation by somebody who gave them a pretty bad name. Usually these are very shy creatures. If you look a little closer, they cannot even fly. They have small wings on their back, but they're too small. Even if they would flap them like crazy, they couldn't lift off. So a very shy, timid creature hiding in the dark, doesn't do any harm, shows up sometimes at night, but would never go after anything or to humans. Some of them actually end up in the Malaise trap. It's a miracle for itself because they have to crawl all the way up, which is quite a distance for small animals like that. The last box contains several orders of insects. All of them have one in common. Their larvae live in water for quite a while. Many of them actually are pretty fierce predators while they're down there. And once they emerge, they turn into completely different creatures. First group, the dragonflies, not very common in your traps simply because of the size. And these guys are so good in terms of flying. They have 4 independent wings. They can maneuver forward, backwards, up, down, doesn't matter. So it's, among the insects, probably the best flyers. The other interesting group here is the caddisflies. So their larvae underwater build their own casings out of the debris. So they build a little tube in which they carry around, pretty much like a snail carries its own house, but they have to build the casing by themselves. And then we have the other 2 groups, the mayflies and the stoneflies, similar lifestyles. Remarkably for them, and also for some of the flies, is the fact that a lot of them actually, once they hatched as adults, so after metamorphosis, they don't eat anymore. Some of them just stop the eating behavior. Others, and there are some mayflies especially, they don't even have a digestive tract anymore. So they have no structure to feed. And it's not necessary because as adults, they don't live very long. Some of them a couple of hours, a few days. The only reason why they adults is pretty much to mate. And then once they've done that, they die. And the largest span of their life they spend as larvae underwater, preying on anything and getting ready for adulthood. So those were these insect groups I wanted to show you. Before I take you up on the little bus journey, a few things to mention. The first thing is, I hope you've noticed and you've heard that we have a blog where you can actually post whatever you find interesting about your trap and what you did in the last couple of days. So a lot of groups have done that already. We would encourage you to continue. It's pretty fun, so you can also post images. There's also something I'm going to talk about later for a bit. We have a Twitter thing going on right now where you can actually post a selfie of you on your Malaise trap. So, as I said, I'm going to tell you about that later on. And we have this book here, which is going to be at the end of this session. So we'll have a little slideshow that shows you all the pages of the books that shows a bit what the BioBus is all about. So this is the BioBus in front, and you'll see it in a minute when I move the camera, which is our vehicle we have for already 5 years, taking it out in the field. So again, that book you're going to see as slides at the end of today's thing. So let me change the camera thing here so it gets a little choppy now. So you're looking at the BioBus. So essentially an RV. It's really technically not a very special thing. But what we did is we redesigned it a little bit so that it's eye-catcher and that people know when we're coming. And then when we attract masses and explain them what we're actually doing. The bus is, as I said, our field vehicle, which means in the summer for the regular season, so we're talking May to September, we drive into several places in Canada. We always pick west, east, mid, somewhere, a region where we can visit a couple of national parks, usually about 10 to 15 of them, where the people can use all those traps I presented to you and the standardized sampling method I talked about. The bus is home for 4 people in the summer. Get this off here. And now bringing you inside and show you what that means if you have to be on that thing here for a month with 3 other people. So let me put you down here. You can have a little look at what the interior is. You can see the bed up there, which is probably the most famous spot for people to sleep in. Yeah, as I said, 4 people. We changed the crew after 4 weeks simply because we know that if you have to live with other people that long of a time, cramped into this small environment for a couple of months, that's probably not so good, and people don't like each other after that. So we changed the crew, and we want to give more people the opportunity actually to go to places where you normally don't go. So as I said, we go to national parks a lot with that. So this year the bus went to Alberta, British Columbia, and the Yukon Territory. I think in 16 different parks, collecting insects, collecting other things as well. But if you're on board on the bus, you give up a lot of your personal space. You're allowed to bring a small backpack with your personal belongings and a sleeping bag, because the rest is pretty much occupied by equipment, supplies, and all that kind of stuff. So you can see our dinner table or breakfast table is not always used for that. Once they're done dinner in the evening, you often sit down with microscope and everything you need in order to do the first sorting of bugs, sorting through the day's catch, in order to have something pre-sorted coming back to our institute. So that's part of the work as well. So as you see, it's a non-RV. It has everything it needs, a little kitchen, freezer and fridge here on the side. And if you look in the back here, 2 beds for the other people, shower, and then washrooms also in here. And then if you're wondering where the 4th bed is, that's actually hiding under the sofa here. So home for 4 people, usually 2 researchers that are permanent staff here, and then we take along 2 students that are in their final year of environmental science, zoology, biodiversity, whatever major they're doing here at the University of Guelph. So it's an opportunity for them to do field work that contributes to whatever we do in research, plus having a good time in places where you usually don't make holidays because it's a bit too far away. So let me take you out of this bus again, put you back on the tripod, and then I'm ready to answer your questions if you still have some. Just 1 second. It's a bit tricky here. Okay. I think we're good. Yeah. No, I'm not out of the picture. Do you have any questions already? No. We'll just give you a few seconds. While you're thinking or typing your questions, I explained that one thing I was talking about. So I said, we want you to make a selfie of you and the Malaise trap. And we have a hashtag, which is SMTP selfie on Twitter. And you can post your image there. What we're going to do is we pick a few, the best ones, and the winners will get something which is called the Life Scanner Kit. And that's this thing here in front of me, which contains 4 vials where you can go collecting for barcoding whatever you want. So you don't need a Malaise trap for that. You can collect something else that fits in there. You need your cell phone because there's an app coming with that allows you to take images of whatever you collected. Takes an image of the vial. There's a little 3D barcode on it, which sort of allows us to connect the data right away to our database. And it also uses the GPS function of your cell phone to give us an idea of where you collected the specimen. So you don't have to do a lot to submit it. And later on, once you've filled them all and the data is on its way through the cell phone, ship it to us. We do the barcoding, and then you get the results back right on your cell phone. So right now, there's sort of the first thing to get the word out there is to have a couple of those for free. So you can sign up on that Life Scanner website, and hopefully you'll be drawn. Or you're a lucky winner with our selfie contest, and you get some of them. So there is a question. Good for me. What role do you think the government should play in protecting biodiversity? That's a difficult political one. More than they do right now. Far more than they do right now. What they have in place is some sort of a regulation and laws that sort of should clearly define what people are allowed to do in order to protect it. So what I'm talking about is mainly how do we how do we use the land we have? So any kind of development for new houses, for resources like mining, oil, you name it. There is a controlling process that has to tell okay, if you take this piece of land and turn it into something else, what is the damage you do? What are the species that disappear? Are these species occurring elsewhere so that there is a chance that they still survive, or are actually driving a species into extinction? There we have a problem because some of these things are not as controlled as people would like them to have, especially as biologists would like it to have, because a lot of industry and also land development is always granted permission without really closely looking at how much damage we cause. And I would hope that our government is putting much more effort in developing that, and instead of taking away money from the people that are there to do this kind of checking, the job there, giving them back money. Currently they save money by taking it away from these kinds of institutions that help us to control ourselves instead of giving them more, and they need more actually. No, there's another one. It's almost Belton, okay. One group has to bail out. Bye-bye, Ross. Any more questions? Come on. Well, if not, usually it's when I start my famous last words, people come in with questions, so I keep talking. So I think I've shown you everything I could in terms of the traps, of the bugs, and the insects you have. You're already well into the program, so you had a chance to see the trap in action. You see what came in there. We were lucky with the weather, actually, so that we had enough higher temperatures that you get a lot of insects in there. So what happens next is something you can actually watch, because what we will do is put a few videos, two at least, up there that shows what's going on in the lab once a bottle of yours arrives here. So what people do in sorting and in DNA process and everything else. So there will be 2 videos at least that show that and give you an idea of what the process is in-house, sort of helping us to bridge the time, because it's taken a little while until we can give you the final reports for the entire program. What we do is we will take not all of your bugs. If you have 10,000 of them, we honestly don't have the money to run them off. But we will take from every school 285. That's our magic number. And pick the ones that look different enough so that you have as many different species as possible. Sequence them, get a DNA barcode, and then hopefully be able to tell you what species it is. That being told, for some of them, I'm pretty sure we cannot tell you all the way down to the species ID what it is, because some of the research is not even there. We could tell you it belongs to that group, and that's as much as we can do at this point. There's a couple of them that are really tiny and all look the same, so there's a lot of research that needs to be done before we can actually tell you this is species X, Y, and Z. But that's pretty normal. Okay, so thank you for being around, and hope you have still a good time with the trap. Good luck with trapping and everything else, and thanks for being here with us for this hour.